Hello, my name is Dr. Tom McGovern. I am your AMP2 lab instructor, and I'm going to start talking about blood in a couple seconds. So, blood is the life-sustaining transport vehicle that goes to every cell in the body of the cardiovascular system. So, knowing that, blood really has three functions. It has a function of transporting, and it has a function of regulation, and it has a function of protection. So transporting is pretty straightforward. It's going to transport oxygen to every cell in the body. It's going to get rid of the waste products, which for the most part is carbon dioxide. In addition to that, it's going to transport nutrients and hormones to all the cells in your body. Regulation is going to maintain a normal temperature, and when you finally get to the urinary system, allow you to have buffers in your system. The protection portion is going to be probably the easiest one to understand. If you have some sort of bacteria in you, you will have white blood cells rushing to it to keep you in homeostasis. The interesting thing about blood is it is the only liquid or the only fluid tissue in the body. If you remember back to AMP1, a lot of tissues were bones and muscles and stuff like that, and then they had blood in it. Blood, because it is a fluid tissue, is kind of unique. So the fluid tissue is the matrix, and that is called plasma. Plasma is a non-living fluid. And then in the non-living fluid, the plasma, are going to be formed elements. There's three types of formed elements. There are going to be cells that are red that are called red blood cells or erythrocytes, cells that are white blood cells that are called leukocytes, and platelets. These cells will be suspended in the plasma and flowing around your whole body. The way that you can find out what it is by something called a hematocrit. The definition of a hematocrit is it is the percentage of red blood cells in a sample. So if they were to take some blood out of you, put it in a test tube, spin it in a centrifuge, what would happen is the heavier components, and that would be the red blood cells, would go to the bottom of the test tube in the centrifuge. Then there would be a top portion, which is the plasma, which is going to be kind of yellowish. And then sandwiched in between the plasma and the red blood cells is something called a buffy coat. The buffy coat consists of two components. They are white blood cells and the platelets. And so if you look at the picture in front of you, if your hematocrit, which is the percentage of red blood cells, is 45, that's probably the best way to remember what is normal. So a hematocrit for males is 47% plus or minus 5, and for females 42% plus or minus 5. If you want to make your life simple, just remember 45. So if you have a hematocrit that's 30, you are probably missing red blood cells and you're anemic. And so, as it says there, the buffy coat is less than 1% because you don't need it unless you have a disease that will have the red or white blood cells rush to destroy whatever's not supposed to be there. And then on top of that, as you saw in this picture, is going to be the plasma. So simply put, 45% is a normal hematocrit, which is the percentage of red blood cells, and the other 55% is going to be the plasma, and less than 1% is sandwiched between the two layers there. More than likely, all of you have had something that has bled, and it does have a metallic taste to it. If you have a scarlet red blood, you have a high oxygen content. If you have blood that looks more like molasses, you're going to have a low oxygen content. The pH is going to be neutral, and so as this graph shows you, it's somewhere between 7.35 and 7.45. For males and females, the easiest way to remember this is it's about 5 liters. For females, they're 4 to 5 liters of blood, and for males, it's 5 to 6 liters of blood. So the blood, as you look at it, and here is a photomicrograph of red blood cells. They're the red things in the background, and also the white blood cells that don't really look white, but they're not the red blood cells. So what you would do with this thing is be able to identify the red blood cells, and the reason why it's important is because it gives you a relationship as to the size. The background red blood cells are going to indicate what size the white blood cells are. In this particular example, if you look down at the lymphocyte, it is almost just a little bit bigger than a red blood cell. So that would be a small lymphocyte. Whereas if you go over to the left, the monocyte is probably close to 4 times the size of the background red blood cells. You will not get to look through this, but it's just so you get an idea of what it looks like. So back to red blood cells for a few seconds. They are in a small diameter, that's 7.5 micrometers, and their purpose is to contribute to gas transport, as we mentioned before. I personally think it looks like a snow tube without the handles. And the purpose for that is for transportation of the gases. And hemoglobin is going to be what's going to bind to the oxygen, and it's also going to get rid of it. So this is the stuff that you'll learn in the lecture portion. We're just going over the lab components in this particular one, but it's kind of interesting. So let's just get to here, because we're not doing anything but this. A lot of you have probably heard of anemia. This is more the lecture than anything else, but anemia means it does not have enough red blood cells. So if you go back to your hematocrit, you would have less than 45% hematocrit. So if you had like 30%, you're pretty anemic. And there's lots of different reasons for anemia. One that most of you have probably heard of is sickle cell anemia. In this particular case, the picture on the left is of a red blood cell, and when it sickles, it's the picture on the right, and it makes it look like a sickle. And so in that particular case, the person who has sickle cell anemia is not going to be having enough oxygen to carry to move around. White blood cells are going to be leukocytes. These are the only cells found in blood that are actually a formed element of a complete cell. So whereas the red blood cell has ejected all of its organelles, and that's why it looks like a snow tube, in the case of the white blood cells, it still has everything inside of it. And the white blood cells, the easiest way that I can teach you to remember it is, there's 2 types. One are going to be granulocytes, meaning that the granules in the cytoplasm are visible. And the other one is agranulocytes. The prefix A means without the granules. They're there, you're just not going to be able to see them. The mnemonic that this particular textbook tells you is, never let monkeys eat bananas. And so that tells you that the 5 different types of white blood cells are N, L, M, E, and B. I'm going to give you another one, which is another mnemonic that uses the same thing. And so in that particular case, granulocytes are neutrophils, eosinophils, and basophils, whereas the agranulocytes are lymphocytes and monocytes. But if you use the mnemonic never eat bananas, which is neutrophils, eosinophils, and basophils, you have NEB. And then the agranulocytes, so if you're not going to eat bananas, who's going to eat them? Let the monkeys. So that would be L and M. If you look at this particular chart here, you can see to the right of each name is the percentage of how many would be in a normal sample. So neutrophils, as it said earlier, are the ones that are going to be the most that you'll find in any sample. And if you easily want to remember it, your lab book tells it from 2/3 to about 3/4%. This one says 50 to 70% are going to be in any sample neutrophils. If you go down to lymphocytes, the second most numerous one is going to be 25 to almost 50%, and those are lymphocytes. So if we go back to never eat monkeys, never let monkeys eat bananas, it's neutrophils, lymphocytes, monocytes. They are the most numerous, and you can see here that monocytes are about 3 to 8%. Finishing up the rest of the 5 of them, eosinophils are going to be 2 to 4%, and then basophils are going to be 0.5 to 1%. So most of the time in any sample you are going to find neutrophils and then at least a couple lymphocytes, but occasionally you might find an eosinophil, a basophil, and a monocyte. The best way to learn all of these is to be able to see what they look like. And in this particular picture here, they look uniquely different. But the red blood cells in the background let you know what size they are. And so as you're looking at that, keep in mind that this background red blood cell is probably double in the neutrophil, or maybe 2.5 times. So the information below each one of those are going to be what you're going to have to remember. We are going to ask you then what they have as a function, and I'll show you a chart for that one. So a neutrophil is multi-lobe, meaning there's more than one lobe. And so if you look at the neutrophil there, that one there has 3 lobes that you can see in the nucleus. And it's probably twice, maybe a little bit more than twice, the same size as the background red blood cells. So that's the first of the granulocytes, meaning you can see the granules in the cytoplasm. The second one is an eosinophil. An eosinophil is unique. I think they say it's bilobed, which means 2 lobes. I think it looks like a bikini bra, just because there are 2 lobes, and the granules in the cytoplasm of the cell look either strawberry red or orange red when you're looking at them. This particular cell is responsible for a parasitic infection. So if you have a parasitic infection and you had your blood drawn, you would probably have as many eosinophils as you would neutrophils. Remember, the eosinophils are only about 3 to 8% in any given sample. So, but they're unique in how they look, and they're about the same size as a neutrophil. It's just the cytoplasmic granules are going to look unique. A basophil is also about the same size as the neutrophil and the eosinophil, and in its particular case, it has a purplish black cytoplasmic granules. Kind of looks like someone has splatter painted all over it, and it is so rare, it is less than 1% up to 1%. It's going to be involved with allergic reactions. So those are the 3 granulocytes: neutrophils, eosinophils, and basophils. And as I said, I use the mnemonic never eat bananas for neutrophils, eosinophils, and basophils. And then who's going to eat the bananas? They are going to be the A granulocytes. And the 2 A granulocytes are going to be lymphocytes. So that's going to be my sample or example of LET. And then monocytes are going to be the monkeys. So if you look at them, the small lymphocyte is pretty much the same size as the background red blood cell. They're very spherical, and to me, around the nucleus is always a lilac rim. And if you look at what it is here, you can see this lilac rim around it. That is a key indication that it is a lymphocyte. Lymphocytes can also be a large lymphocyte, which would be double and probably the size of a neutrophil. But it would still have that purplish rim around the nucleus. And it's going to be involved in immune responses. And a lot of you have probably heard of lymphoma and stuff like that. That's where those leukocytes are going to be all over the place. The monocyte is the one which would be, in my case, the monkey representation of the mnemonic. It is very, very large. It is probably 2, 3, maybe up to 4 times the size of the background red blood cells. It does have a kidney-shaped nucleus, as you can kind of see in that picture. I think it looks like a boys' room, bathroom, toilet seat. You have to go to the boys' room to see that. I don't suggest that. But the background cytoplasm is that pale lilac color, very similar to what you can see in the lymphocyte. And the interesting thing about a monocyte is if you want to think of this as someone who is going to hunt out the bacteria and kill it, a monocyte can actually change itself into a macrophage, leave the bloodstream, and specifically go into certain organs. For example, in a kidney or in a liver, it's called a Kupffer cells. So the monocyte has left the bloodstream, gone into the liver, and it is now hunting down the bacteria or viruses or anything else that's going to be involved with that. So each one of these will just be a close-up picture of what that is, a close-up picture of a neutrophil. Remember that the key features of it, it is about double the size of a background red blood cell, and it has a multi-lobe nucleus. In this particular case, it has 3. It does have red and blue cytoplasmic granules, which makes it look almost purple. And that's the one that you would find most often. And you know, most textbooks have them like anywhere from 50 to 75% in a normal sample. Eosinophils are going to be the second one I'll show you. Once again, this particular text shows it as it resembles an earmuff, and it's going to have red stained granules that contain digestive enzymes that are going to eat things like parasitic worms. And here's a picture of what it looks like. Bilobe nucleus, in this particular case, the red cytoplasmic granules, in this case they look kind of strawberry red, but sometimes they look almost orange red. Basophils are the rarest. They are going to be around the same size as the neutrophils and the eosinophils, but they're going to be very, very dark purple, dark black in color, and it's going to contain a lot of histamine. So in that particular case, if you have a histamine reaction, that is what's going to rush to the area. And if you look at the picture here, it does, if you look, you can see the background red blood cells, but it just looks like it's speckled with splatter paint all over it. Purplish black cytoplasmic granules. The agranulocytes, remember, these are not going to have visible cytoplasmic granules, even though they probably do. It's just you're not going to be able to see them. A lymphocyte is going to be the most second numerous after a neutrophil, and it's going to be found in lymphoid tissue. Most of you have probably dealt with lymphoid tissue in the form of lymph nodes. So if you have a terrible sore throat or a streptococcal infection in your throat and you get an enlarged nodes in your neck, it is because lymph's purpose is to filter out the bacteria before it sends it back to the heart. So in this particular case, this is a small lymphocyte, and the only reason they call it a small lymphocyte is because it's just a little bit larger than the background red blood cells. It is very spherical. It does have a lilac rim around the nucleus, and that's very indicative of a lymphocyte. If it was a large one, it would be maybe double that size. Monocytes are the largest of all the white blood cells, and you got about 3 to 8% of them in any sample. They are going to contain a deep purple U-shaped or kidney-shaped nucleus. They have the ability to actually leave the bloodstream, the circulation, enter the tissues, and differentiate into a macrophage. They are going to hunt down anything that's not supposed to be in your body, like viruses, like bacteria. And so this is what one of them would look like. It is a monocyte, and it is kidney-shaped in this particular one. You can see the lilac rim around the darker purplish blackish nucleus, but they are much larger. And you will have the pale blue cytoplasm surrounding it. The unique feature about the monocyte is it can change into a macrophage and hunt down and kill. We don't use the word kill, we're going to use the term phagocytize, which means eat or kill the bacteria. And so here are all of them lined up together. So as you look through them, Never eat bananas, neutrophils, eosinophils, and basophils, and if they're, that'll help you remember for granulocytes. But who is going to eat the bananas? Well, let the monkeys, and that would be a lymphocyte and a monocyte. So if you look at them all next to each other, these are perfect pictures that you probably would never find in the sample that you would find in lab. Platelets are the other formed element that you'll have. Platelets' main function is to form a temporary platelet plug that will help seal any break that you might have in a blood vessel. If you have ever cut yourself, even a paper cut, you are going to have platelets rushing to the area to make sure that you can stop the flow of blood. So platelets are interesting because they come from a hematopoietic stem cell, and then they just kind of explode apart as you look on the right here, and they become part of the blood, and they are just floating around in the bloodstream looking for some break in a blood vessel, and they're going to help seal it up. So here is, in this particular textbook, a collection of what you're going to be asked about for the practical. So if you use at the top the red blood cells, they are binucleus or biconcave, anucleated. The reason I think that they look like a snow tube is because they have already ejected their nucleus, and it made it a little bit hollow in the center. They say it looks salmon colored, but it looks kind of reddish color. You have a lot of them, 4 to 6 million of them in a cubic millimeter of blood sample. If you look at the function, it transports oxygen and carbon dioxide. Those are gases. And so remember, that's what you're going to use in the background as the reference size for the white blood cells that are at the bottom of this page. Leukocytes, or white blood cells, there aren't too many. There's 4,800 to about 10,500. But in that, they're going to do a lot of other things. So this chart shows you what it looks like on the left, its name, then it tells you a description, multi-lobed nucleus, and then on the right side, it tells you what it does. Phagocytize in English means eat bacteria or kill bacteria. And so they're the ones that are most numerous. If you remember, that's about 50 to 75% in any sample. The second one of the granulocytes is the eosinophil. Once again, it's going to have the reddish cytoplasmic granules, or orangish. Bilobe nucleus, they are going to have as a function an ability to kill parasitic worms. Where would you get parasitic worms? Well, a lot of times if there is not really good cleanliness in your food, and I'll give you a gross example. If you have parasites in your lettuce and you don't wash your lettuce, even though it says it's been washed 3 times, you can then ingest the parasitic worm. And so in a lot of countries where they don't have a lot of clean water, they get parasitic infections. And so they would have a lot of eosinophils because of that parasitic infection. The last one is the basophil, once again, of the granulocyte, a bilobe nucleus that you can hardly see because of the purplish black cytoplasmic granules. It's going to be responsible for histamine and other mediators of inflammation. And so if you've ever been stung by an insect or a bee, these are the guys that are going to make it itch as you're healing. So this one has now gone to the agranulocytes. Once again, at the top for illustration is going to be the red blood cell. Once again, it's going to transport oxygen and carbon dioxide. Also nutrients and hormones will go throughout the body. But underneath it is the agranulocyte. So in my mnemonic, remember, don't eat bananas, let the monkeys. The lymphocyte is going to be this perfect one there, has a very oval looking nucleus with a lilac or kind of bluish halo around it, which is going to be the cytoplasm. Now you really can't see any granules in there just because of the magnification. So with a lymphocyte, if you look over to the right, it's going to mount an immune response by direct cell attack or through antibodies. So that's its job to, and if you have a high lymphocyte collection in your sample of blood, remember it's the second most numerous after the neutrophils, and this is about 25 or up to 25 to 40% that you would find in a sample. That would concern a doctor because they might have to find out why do you have so many leukocytes in circulation. The monocyte is shown there very gigantic. It is monstrous in size. So it is 3, 4, and even larger than the background red blood cells. Its purpose is to eat bacteria. That's what phagocytosis means over on the side. But it also can not only find the bacteria, it can develop and change or morph itself into a macrophage in the tissues and hunt it down and then eat it wherever it had to go. Sometimes there's monocytes in lung tissue and in liver tissue, and the monocytes are now going to be changed into a macrophage, and they're going to kill and eat bacteria. At the bottom here is the platelets, and they just kind of show it as cells that kind of have burst apart, and their function is to find small tears, because remember it's floating all around your body, and it's going to be instrumental in blood clotting. So if you just want to remember blood clotting and platelets, that's probably a good way to think about it simply.